Good afternoon. Thank you for joining us for today's COVI Lecture Series presentation. This afternoon, we are excited to welcome Dr. Charles Després, COVI Fellow and Professor of Biological Sciences here at Brock. Charles is an expert molecular biologist and biochemist studying signaling pathways involved in plant immunity with over 20 years of experience in the field. He is known internationally through his paradigm-shifting discoveries published in high-impact journals. Charles has received numerous prestigious awards, including a Tier 2 Canada Research Chair and the C.D. Nelson Award from the Canadian Society of Plant Biologists. Please join me in welcoming Charles.
Thank you very much, Andrea, for the invitation and the introduction. I hate starting this way, but I have to apologize for the title. I think I'm not going to talk as much about it as I would have liked when I sent that title, but you'll see some of it coming through the seminar. And since it's my first time giving a seminar as part of COVI, I thought I would try something quite different, and I hope if you're not happy, throw eggs, but no tomatoes, please. And I'm going to be fine with that. So I'm going to talk a bit more about myself and as I move through my career. And the idea is to give potential collaborators maybe some sense of what I like to do and what I can do if I collaborate with them. And it's really in that sense. And some of the things that I do today that actually don't relate to plant at all, but they still give an idea of what I can offer. So part one, I'll talk about who I am, and really it's in the perspective of maybe collaborating with people. In the part two, I'll talk about something I've done at Brock, which is the characterization of an important salicylic acid receptor, which is involved in plant immunity, a special type of immunity called systemic acquired resistance. And from then, I started really have all the tools and all the ideas, or so I thought, to move into more applied research. And really since 2012, that has been my main interest one way or another. So part one. And for me, really everything started in 1985. I was 16 or 17. And I used to like to shop for used books. And I found this book called The Biological Order. And it was from a Nobel Prize winner, André Lwoff. And the book who had won a Nobel Prize along with Monod and Jacob. And so Lwoff got the Nobel Prize for working on viruses. But you may or may not know, but Monod and Jacob were more popular or famous for their work on the lac operon. And Lwoff talked about his work, but also the work of Monod and Jacob, and it's really that part that interests me. And I really realized back then, in high school, you know, you look at microscopy slides and you see structures, but you don't really link that to living necessarily. And for me, that was the realization that cells could be looked at as machines, and they had functional parts inside that could be understood like an engineer or reverse engineer process would look at things. So that was what really sent me on my career. So in 87 to 1990, I did a BSc at the Department of Biochemistry in the medical faculty at the University of Montreal, and you'll see a trend here that there's a definite lack of plant background in my career. So back then, during the BSc, I became aware that transgenic organisms were a thing, and in the early 80s, there were reports of transgenic mice and transgenic tobacco coming out. Mice were a bit earlier, but really transgenics with real functional genes were about from that period of time. And for me, sorry, one of the important moments during the BSc was my visit to a lab where they were doing lots of transgenic plants. And I asked a lot of questions about what we can do with those, and I realized that we could actually, they were ready to introduce foreign genes, and that opened up the idea that you could study really genes in a whole organism. In animals, it was still the use of cell line, and it's still the case today. And rats and mice were not all that, at least where I studied, it was not very prevalent. So the plant looked like a good system to work on. So in the summer of '89, I decided to do an internship, and I did a lot of tissue culture, generating transgenic plants, and it was a system, potato system or wild potato system, and I did also something called in vitro tuberization, where you can produce mini tubers, mini potatoes in vitro, and that came in useful during my PhD. So 1990 to 1992, I did an MSc in molecular biology, and this was at the Cancer Institute of Montreal. Again, not that much plant there. So but my lab component was at the Botanical Garden. You have a picture of Montreal, picture of the Chinese or Asian garden, at least, with the mass of the Olympic Stadium in the back to break the harmony there. But during all the lectures at the Cancer Institute, I became really familiar with signaling in animals and gene regulation and a bit of clinical trials and so on. But the main project was helping a PhD student at the time, and he was doing a map of potato genome, so a linkage map, so that breeders could use it to introduce or introgress traits of interest in the potato. So lots of RFLP, which is something that probably does not exist today. It's a bit archaic, but that was cool at the time. And also my main project was on a system called self-incompatibility. And so I did also some cDNA libraries, which were a fun thing to do at the time. And I was just at the beginning of the PCR, and these were interesting machines at the time where they were water cooled. They would hook to the tap, and you would run cold water through them. And whether you work in summer or winter, you would have different protocols for the water temperature. So lots of fun then. And yeah, studied relative of wild potato in the context of self-incompatibility. So in 92, 98, I did my PhD at the back to the Department of Biochemistry in the Faculty of Medicine again. And really what I wanted to do there was to bring the study of transcription factors, gene regulation, and there was an opportunity to do so in a lab there. And that was in plant, potato was the system, and I'm going to show you some of the results that I got during that time. So here's a slide, one of the many things that I've done, but this was a promoter deletion analysis. It was done in transgenic potatoes, and I would use microtubers to study the system. And in that lab, they had a gene that was inducible by an elicitor that is found in a pathogen called Phytophthora infestans. That elicitor is a fatty acid called arachidonic acid, which is generally not found in plant, maybe not found at all in plant, I'm not sure. But it was found in the pathogen, and certainly in animals, it's an important mediator. Inflammation, for example. So the promoter was cloned, and you can see some of the first construct here. And I think there was a problem with that slide. It's okay. I had a longer version of the promoter, but it's cut. It's not a big deal. So here you have a promoter, like 670 base pair, fused to a little bit of the protein sequence of that gene that is now called PRTNA. and fuse to a reporter system, the beta-glucuronidase gene or GUS gene. And then I would take these mini tubers and slice them in half, one treated with water, which was a control, and one treated with the elicitor, the arachidonic acid. And the idea was to find regions in that promoter that were responsible for the induction by the elicitor. So that's what we see a little bit here. There's a deletion at -70. You can see in black the elicitation, the level that you would get after elicitation. In the gray bar here after wounding. And here is just if you take an intact tuber, you would have a certain level of induction. So you can see that it's inducible by the elicitor. And the idea was to make all these constructs, make the transgenic potato, generate the tubers, and test the promoter. So you have a series of deletion here. And at about -135, there was a big drop. We don't see it on the picture, but there were some indication that it was still inducible by the elicitor. So, and we knew that there might be some repressing element further downstream. So a construct, the -135, lacking an internal area where a repressor could bind, was removed. And then you could, if you compare the -135, it looks like nothing is happening. If you go back to this one with the internal deletion, it was still inducible by arachidonic acid. And at the end of the day, a region was mapped between -135 and -105 that appeared to be important for the induction of the gene by the elicitor. So, and we published something in the plant cell in 95, and then from 95 to 98, I started the purification of that factor. I had the DNA element, so now came the purification of the factor. Took many years to do that, something that is that is not done today much with all the genomic information, but that was a different story back then. So I use a technique to follow the transcription factor on the piece of DNA called the electrophoretic mobility shift assay, or gel shift, or EMSA. And here you have a crude extract, and you have two bands binding to that particular piece of DNA, so one or two factors binding. And then after many years, I figured out a way to purify the factor. So one was an anion exchanger, followed by 2 rounds of DNA affinity. Essentially, it's the piece that you use in the gel shift that was coupled to a solid support, and 2 rounds of purification through that gave an almost pure factor. And in B, you can see one of the purifications. This is a silver stain. And you can see in a crude extract, you have many bands, many proteins. After 1 round of affinity, it drops quite dramatically. After the second round, it was mainly 1 band, but some shadows of other things. And in the last link was a cross-linking experiment where you radioactively label your piece of DNA. You shine UV light, you have the DNA cross-linking to the protein, you run this on the gel, and that allows you to verify which protein, which molecular weight contains the binding activity, and it corresponded with the band at 24 kilodalton, so that name, people started calling that factor P24. And so that was part of another paper, and that's about the time where I finished my PhD. It was long enough, time to move on. But other people stayed behind, continued the project, and one of my colleague, Alexandre Joyeux, went on to get the cDNA clone for that transcription factor. Underlined here were the peptides that I obtained from purification and microsequencing of the protein. And protein is not so interesting today, but just to show that the step that were used. And later on, one of my colleagues, Daryl Devo, who is now a professor at the University of Toronto, and this I didn't participate in that aspect, but I thought it's important, went on to crystallize that P24, and you can see the crystal structure here. Looks like a beautiful protein to me. It's actually a tetramer, so you can see in the different color, the 4 units forming the complex. So for me, that was an interesting journey. And so I went from a gene identifying a regulatory element, characterizing or purifying the transcription factor and participating in the cloning of the gene and the crystal structure. So when I reflect back on what I did, there were some positive, some negative, and some of the positive was really being part of that pipeline was quite interesting and enlightening for me and motivating at the same time. And if you would be, having spent a lot of time with medical people, having had a crystal structure, that would have mean, okay, now you can go for a drug, engage into a drug development pipeline and so on. But there were things with the P24 that were not all that palatable. It felt like it was a transcription factor really at the end point of a cascade and not really a master regulator of immunity. So for the postdoc, I really wanted to look for something where higher up in the signaling cascade that would be more of a master regulator and had more potential for commercial application later. So I moved on to what was called the Plant Biotechnology Institute, or PBI at the time. And I was there from '98 to 2002. It's now called NRC Saskatoon, one of the branch of the National Research Council. And my motivation to go there was, first, they had good salaries there compared to the time as a student. So anything is a good salary compared to what you get as a student. So I don't know if that's a big concern. But joke aside, it was, I saw this as a place where I could still do basic research, but because it was a crown corporation and they were quite involved in applying research, I thought it would be a good way to do what I know how to do, but to have to peep at what is being done in the realm of application. And about that time, just before I finished my PhD in '97, there was the report of a cloning of a gene called NPR1, and that indeed was a master regulator for plant immunity that is dependent on salicylic acid, that is called systemic acquired resistance. So that was interesting, and there was someone there, Pierre Faubert, that was actually starting a project on that, and it felt really like just impeccable timing. And what I did not know at the time is that I would bring that project to Brock, and I would continue it as a faculty member. So that was also a good bonus, and I worked on that until about 2012. So second part, I want to show you one thing that to me was important and really put all the ducks in a row for me to start applied research. And it was really the characterization of NPR1 as a salicylic acid receptor. And the idea of the two being connected was nothing new, but no one had really shown that NPR1 is actually binding to salicylic acid. And there were some reasons why as well for people not being successful in that area. So just a quick recap, in case you don't know what is signal transduction, and this is just a crude cartoon. We have a plant cell here, and any cell, plant cell included, they perceive external stimuli, generally through molecules called ligands. These ligands generally interact with receptors. These receptors are generally located at the plasma membrane, but sometimes also intracellular, and that's the case for NPR1. And then once the receptor interacts with its ligand, it changes conformation, and generates a cascade of events inside the cell called the signal transduction pathway. And not always, but normally, or what's interesting for me, is that this leads to gene activity modulation. Genes get upregulated or repressed, and that this combination leads to an output from the cell, so the cell responds. In the case of signal transduction during systemic acquired resistance, it's a little bit more complex. There are more cells involved, but this is the general idea. You have a first gland that detects somehow a pathogen. This is done through a molecule that can be called an elicitor or a virulence factor. And generally that first cell will undergo cell death, and some signal will come out, will be generated by that cell. It's perceived by the neighboring cells. And in the case of that particular immunity, you have an increase, not in all plants, but an increase of a buildup of the concentration of salicylic acid, or a buildup of the sensitivity to salicylic acid, one or the other. And that is dependent on a gene called NPR1. And this process leads to local resistance and pathogenesis-related gene expression, and one of the marker gene in that system is called PR1. And from that local area, you have a mobile signal that is distributed throughout the plant, and essentially in the distal tissue, in the systemic tissue, and essentially it's kind of a mirror image of what's happening locally, and that leads to the deployment of the systemic resistance. So one of the reasons that it took so long for NPR1 to be recognized as salicylic acid receptors is that all the classic assays that rely on non-equilibrium, because they're quick to do and easy to do, simply did not work. I've tried it myself. And then I remember something from an old textbook from my BSc, a technique called equilibrium dialysis. And as the name implies, it's a technique that relies on the equilibrium of the system. So, and that was the key to the success. So in a nutshell, you have a vessel, you have a dialysis membrane, and the dialysis membrane, so generating two compartments. The dialysis membrane allows small molecule to move freely across the membrane. So essentially, membrane is invisible to these small molecules, but larger molecules like proteins, they are contained within their partition. In that case, you call it compartment A, and what you do is you put radioactive salicylic acid in one of the compartments, the free unbound protein in the other compartment. You wait a certain amount of time until equilibrium is reached, so the assay will distribute equally through the two compartments, will bind or not, but in that case will bind the NPR1, and then you can calculate a dissociation constant. So the square box of the free salicylic acid. The NPR1 complex to assay can be, they're in chamber A, but there is also some free assay, and the equation is that you calculate the radioactivity here minus what's in compartment B, because for the free assay, the two compartments, there's no barrier between the two compartments. So you can get the bound NPR1, and then you can get the free one by looking at your initial concentration of NPR1 minus what is complex. That gives you the free one, and this is just a regular equilibrium or dissociation constant. So the complex in equilibrium with the free NPR plus the free assay. And you can calculate a dissociation constant based on that. So for today, it's not so important, but that was a method that was absolutely required for to uncover that NPR1 actually binds salicylic acid. So lots of things on the slide, but panel A, essentially, I could not see any difference whether there's protein or not in the binding of salicylic acid if I would use the standard non-equilibrium method. But once I started using dialysis, then you can see it. You could see a signal for NPR1, which was not there for a no protein or other controls that has been done as well. And here in C is just a graph of a typical rectangular hyperbola function that you obtain when you do receptor-ligand interaction. So it looked fine, and you can calculate a KD and a Bmax. And that was a demonstration, finally, that NPR1 is indeed the receptor for salicylic acid. It's not just somewhere in the pathway, but it's important for the interaction with SA. And here, where I think it becomes interesting, I started looking at the lab started looking at some of the specificity of the NPR1 assay binding. And here, the assay is still based on the equilibrium dialysis, but it's a competitive experiment. So you have labeled assay, and you throw in some cold molecules. So you start by throwing some cold assay, so you compete the hot assay with cold assay, and that gives you the standard inverse sigmoidal curve as the percent of radioactivity bound to the protein decrease as a function of the cold competitor. And you can see SA competes SA, that's the standard where you start. And then there are some molecules that essentially don't compete or don't compete very well. And there was something interesting here. There's a molecule called BTH. And that BTH is an active ingredient in a commercial product called Bion or Actigard, depending on your country of origin. And this was a compound that was developed using the NPR1 system, but without really knowing that NPR1 was the receptor. It was developed by, I guess, Syngenta or Ciba-Geigy before. I can't remember, but Syngenta was the one owning it later. And so it started being interesting in the sense that you had a receptor, NPR1, a molecule could bind to it, salicylic acid, but also a commercial entity, commercial molecule could also bind to NPR1. So in a way, there was kind of a proof of concept that you could start a drug screen with NPR1 and find molecules that could stimulate resistance in the field. BTH was such a molecule, but it's not very effective. So there's not much usage for it in that sense. So people still use traditional fungicides instead of treating with BTH. It's a surer thing. And there are some other chemicals that do compete or don't compete. Some chlorinated derivatives do compete well, and when you start removing some bits and pieces from salicylic acid, the competition drops. And here's just a bar graph to show that molecules that did bind to NPR1 did also activate this marker gene PR1, which is used as a proxy for the deployment of salicylic acid. So all looked good. And finally, the model, I skipped a lot of things, but the model was that, still is, that salicylic acid bind to NPR1. NPR1 changes, we verified it changes conformation. So it's shown as opening up. This I don't know, but it changes conformation. And the binding of SA, importantly, was dependent on a metal, namely copper, and it was binding to two cysteines, 521 and 529. And that was important for all of these components were important for the binding of SA and the functionality of the system. All of this was done in Arabidopsis. I forgot to mention So for my postdoc, I switched to Arabidopsis and continued with that. So for me, all the bits and pieces were there to start thinking about applying the NPR1 system to some potential commercial application. So what I was pitching to people at the time was that we have problems. We're overusing fungicides. Fungicide resistance is developing in the field, and even not just in the field, but you can see that overuse of fungicides in agriculture is leading to problems in the human treatment as well. The same kind of azoles are used, and resistance is building also for pathogen in the human pathogen. And I've read somewhere that some fungicides, they just become obsolete after a couple of years because resistance mounts quickly against some of them. They're often just derivative of a core structure, and that doesn't help. So that was the pitch, and I would pitch that I have a solution. We could use NPR1, develop a molecule that could stimulate immunity in the field, and the idea would be that people would not have to use as much fungicide. They could scale back on the application of fungicides, and that would increase their time to obsolescence. And so the company that invests in fungicide have a longer runway with them. And in the back of my head also, maybe we could find a molecule that binds to NPR1 that could be less environmentally detrimental compared to fungicides, but you never know when you start. So in 2012 and then to 2017, I co-founded a company based on that idea. It's called Agropeutics, from agrochemicals and therapeutics, with a little tweak at the end of it to make it cool. And I realized I became CEO, and as a joke, I call it Chief Everything Officer, because you have to take care of so many things you had not planned, didn't have to do the day before you incorporated, and that wear me out a bit. Started wearing a suit, which I really never do, and started to shake hands, and I raised some cash, a few $100,000. So the pipeline for this is a bit as follows. The slide is very crowded, and it was based, the drug discovery pipeline was based on what you would really do in the pharmaceutical industry, with the exception of greenhouse and field trials, which would be replaced by some preclinical and clinical trials and data. So normally you start at the target discovery, so you have to find something to drug. And in our case, it was done. It was NPR1, and with a proof of concept, there was the BTH. So BTH is what we call first in class, and I thought it would be smart enough to get something best in class, and everything would be good, so that was the idea. So we started developing an assay for a high throughput screen. And then the idea is to, from your target, you get some hits that binds, and from the pharmaceutical industry, you want a certain number of positive there, and then it decreases as you play with your structure, modify it, and so on. And the idea, I focused a lot on Sartoria blotch and Asian soybean rust. There were lots of reports about the Sartoria blotch having resistance to a number of fungicides, so it felt like a good place to go. And then after that, the idea was that once you find something, it works in the greenhouse, works in the field, then you're in business. With the multinational will take your thing and move it to a commercial product. So basically, at some point, I ran out of cash. And so about just before the high throughput screen, we just ran out of cash, and so it just I closed shop. And just to show you some of the things that we've done there, some of the assay, I think it's important that you know what I can do. So at that point, we had developed a single tube assay, and we were in the business of scaling this up into things like 96 well plate. And then when you, just from the drug industry, pharma industry, when you do high throughput screen, you try to get a hit rate of about 0.05%, and that seems to be the sweet spot. You don't have too many false positives, and you don't miss too many things out of that. So that's kind of the golden rule, and that's what the assay was aiming to do. And then once you get some hits, you have to reconfirm them using the same assay, and then you need an orthogonal assay, so something that is different to confirm that in different systems you see the same thing, so to eliminate the possibility of some kind of bias in the assay. And we were developing at the time our calorimetric skills. So we set on what that was one of the potential assays, was a thermal shift assay. And essentially what you do is you have a protein in a vessel, you increase gradually the temperature, a bit like a PCR machine. And then this is done in the presence of a dye that is relatively hydrophobic. And as the protein unfolds, the dye binds to the hydrophobic patches of the protein, and that changes the quantum yield of the dye, and it can fluoresce at that point. And so that's what we see here. We see the fluorescence as a function of temperature, and you can see that the fluorescence increases up to a maximum, and then it decreases because the proteins start aggregating, and the assay is no good at that point. But you have an inflection point, if you remember your math from some years ago. You have an inflection point here, and you can do, by doing the first derivative, you can get the tangent, which is your maximum. That's what you get here, and you can monitor your melting temperature. So the point at which the molecule is 50% dissociated, 50% folded, 50% dissociated. So this was an assay with NPI1. It's a bit unusual, but normally as you add a ligand, the temperature would increase, the melt temperature. In this case, it decreased, probably because the protein was unfolding already as you add SA. But in green, you have the melt curve for NPI1, and then when you add SA, you have a change in the melt curve. And if you do the derivative of those things, you can look at the peaks, and they were actually separated. So that was an assay we were really considering, where you can monitor the binding to a change in melting temperature. So some of the lessons that I've learned from starting that company is that if ever we have students here, if ever you're in the business of starting something, I think it's a good idea to have some revenues, even if they're not really related to the core of the business, because as soon as you incorporate, money starts flowing out, and so it's good to have a bit of cash flow. And when you're just bleeding when you start the company, so it's best to have a bit of revenue. And one thing I realized is that being best in class may not be the best thing to do. It depends on the field. It's best to be first in class, first on the market with something that works, rather than trying to better something that is already there, even if that something is not that great. And one of the things I realized is that the molecule I was trying to bring to market, to commercialize, it was seen as competing against fungicides, even though it was meant to maybe lengthen their lifespan. It was seen as competing, and I just never got past that. And one thing I realized is that hundreds of thousands of dollars, if you're a student, yeah, it looks great. But once you start spending all that money on everything, from renting space and all that, it just disappears very quickly. So I think it, in some cases, may be okay. But if you can think of something that would raise millions of dollars, it's just better. And can you do that only in human health? I don't know. Maybe not. It's certainly easier there. And the amount of time you spend doing all sorts of nonsense is quite, you just don't see it coming. And for example, we were filing patents, and every quarter it was a fight with the patent office to justify why your discovery is unique and there's no precedent for it, and so on. Sometimes it's just not pleasant. So it's good to have a partner and you can dump some of the nasty stuff on them. So this is a bit of a joke, but after agropeutics, I really started thinking about what I'm going to do, and just going back to my teenage years, I really liked an artist called Klaus Schultz. I don't know if anyone knows about him, but he was one of the first person to start music with synthesizers, and it was really meditative music with long arpeggios and so on. And I thought this music is really good for meditating, and in that sense, it's a bit brain altering. It just plays with your mind a bit. And at the same time, I was quite interested in these psychedelics. I always thought that because they play on your brain, that they might have interesting application in terms of discovering how the human brain functions. I didn't think they would be medically relevant, but this appears to be changing a bit. And during that time, I looked a lot at, from my background, I started thinking of all sorts of things, and I started looking at clinical trials, how they're run, and started looking at especially the mental health space, mental disease space, and started looking at actually the role of psychedelics. MDMA was one of the, and ketamines were some of the ones that were progressing through the clinical trials, and now you have others as well that look promising. So all this, in a way, paid off. A few years later, I became a scientific advisor for a company that develops drugs in the mental health space. And also currently, we're developing some assays for the company. So it's a cell-based assay, special sauce receptor, combination of receptors, and we're developing an assay for that. And the assay is essentially kind of done, and we're looking at miniaturizing the assay. So go from single tube to potentially, this is just a generic picture here, just to show that we're at the step of going into multi-titer plates. And now, maybe for the more interesting part, post-agropeutics part 2 and the grapevine side. I can't remember how it happened, but I started talking with Dr. Jim Wilworth, that is also at CCOVI. And Jim had an interesting system. He's studying cold hardiness, and he had also a molecule that, when applied to the grapevine, could render the grapevine hardier. So that was interesting because it looked like a drug screen could be developed for something like that. And it also removed some of the issues I have with the disease resistance, which is you competing with fungicides. Here, to my knowledge, there's nothing or that you can really use to turn on the hardiness of plants, and I might be mistaken on that. But at least when I got the idea with Jim, there was nothing really commercial, except for breeding and so on. But breeding can take time because you may have an elite cultivar, and then you have to introgress something of interest, but also lots of things that you don't want as well, and it does take time. So having a molecule in the field that you could spray when you see trouble coming, that could be beneficial. So Jim had a system like this where essentially you have a plant, you have a ligand, a molecule, it's presumably interacts with a receptor, turn on some gene, turn on cold hardiness. And can't remember, 2019, 2020, I think, we got funding for 2 rounds of OGWRI, and we were essentially looking at this system. So we had 2 cultivars, one that is grapevine, one that is cold hardy, one that is less hardy. We have our control plants treated with that synthetic molecule that Jim has, same thing with the less hardy, and we would look at some marker gene that came from looking at what's happening in Arabidopsis. And so we looked at some of these marker genes during acclimation, deacclimation, and reacclimation. And so we did this with qPCR for these first 2 years, and the preliminary results are, it doesn't look like the marker genes or the pathway may be identical to what you will see in Arabidopsis. There seems to be something different here. So our next step is we've applied last year, I guess, waiting for an answer for the positive one from the Canadian Grapevine Certification Network. And the idea is to not use qPCR, but to use transcriptomics to look at the pathways that may be involved using these treatments, looking at the pathways that may be involved in this hardiness, cold hardiness. And then there are many things that can come out of this. I don't know if grapevine breeding is a thing, but you could use some of the markers to assist in that breeding or assist for selection. If a grower, for example, is interested in 2 cultivars and maybe would like to know which one may be more hardy, potentially markers could help giving an answer there. But for me, the interesting thing is that having these maps would open up the discovery of a target and potentially the discovery of a drug against that target, or that stimulates the target. We don't even know if it's stimulate or not, or inhibit, but to play with the target and turn on the cold acclimation kind of on demand. So if I go back to the pipeline, and I don't want to go in all of the details of that pipeline, Jim and I would be located right here. We're in the process of discovering the target. But if we get a target and confirm it, my guess is that maybe Arabidopsis come into play here for the transgenic and some of the analysis if it's translatable between the 2 systems. And there's a chance that we could discover some natural product, maybe in a drug screen, and that would lower cost to commercialization. There might be some biologics that are something to think about if you could treat with some modified RNA molecules. There might be option to turn on the system, and I'm just speculating here, I don't know. It depends on what these targets would look like. And the idea would be to bring this to some commercial with assistance from partners. And I realize that the grapevine community or industry is quite small, so realistically, it's probably not feasible to bring something like that to market. But if the compound can be used in many crops, especially some of the big crops, the cash crops, there might be a way to raise enough fund to move that forward and then benefit the community as well. So just conclusion, my interest and expertise, and again for collaborators, and I said after reflection, but you can realize that I'm not really a real plant biologist. I've done no studies on plant. I've worked with Arabidopsis and potato, and I understand and can work with these things. But whenever I go outside, like the grapevine system, I have to work with a collaborator. In my case, it's Jim Wilworth. If it's another system, I need collaborators. It's not something I can do on my own. And I cannot pretend to understand what's going on there. I can understand on a high level breeding, mapping, and marker-assisted selection. And again, I can go and talk to a breeder. I cannot do his or her job, but I can understand what it's about. Really, my expertise lies in the study of receptors, signal transduction, gene regulation, and all the techniques associated with that. Hopefully, I convinced you that I can develop assays for some drug screen. And we're now, which is something that is kind of new for a number of years, but still new for me, we can do all the transcriptomic generation of RNA to sequencing, forming it out, analyzing the data, and we can do this in plants, humans, rats, so forth. And again, I'm comfortable, I think, working with both systems. It sounds a bit odd, but I kind of don't necessarily have a preference. I have a tool set, and this is what I can use. I cannot pretend that I can go really beyond that. So hopefully the seminar was okay, that you enjoyed it. It's different from what I would do normally. I thought it may be useful as an intro seminar as part of COVI. So, thank you. And I can entertain some question from real people and virtual people.
Gordon from the library. Cold hardiness is a problem for Greek vines, Phalaenolian viticulture, and you suggested it's of interest to other plant groups, maybe even human health as well. Is that a response to global warming, or has this always been a problem?
I mean, it's always a problem, and to some degree, global warming is kind of changing the problem's perspective, where you can grow potentially further north as it heat warm up and decrease what you can do further south. You're challenged with drought in the south, but you can expand it in the north. But also global warming is not just temperature, it's about unpredictable weather. So, like cold snap when you never had one, and this may be where having some tools to mitigate that would be great. You could always say, I'm not going to grow anything past this line, but if you want to go past this line, you have a choice of breeding something that can pass across the line, hoping for the best, or maybe having tools, additional tools that allows you to grow into places where you normally cannot grow, but having the tools to still protect the plant. Like you have fungicides, you don't say I'm not going to grow plants where there are pathogens. You're just going to use tools to eradicate the pathogen or control it. This is kind of the same idea.
Thank you.
Aside from grapevines, do you see another target catch crop for this technology?
I mean, everybody uses that jumps on that bandwagon, but population is growing, food shortages are on the rise. So if you can expand in any direction what you can grow, I think it will be valuable. If this actually becomes a concrete thing, population growth, is it like when people invest in the stock market, they always think it's just going to go high up and it never goes down. and it's possible that population just stabilizes. But if it continues growing, there will be pressure for that. I can see where I live that we're ripping out farmland to build houses. So that puts pressure. And so it's just another tool. Any crop has a line where it cannot cross. And with having developing tools, whether it's through breeding or chemical tools, if you can cross that line, then you can grow in places where you could not grow before, and that means potentially more food. So I think it's applicable to anything. If you want to turn this and bring it to market, I think you have to look at soybean and rice, and they might have different problems where salinity is an issue in some countries more than the climate. So that's not my thing to look at salt tolerance, although we do have a project on that, but I'm not sure it's going anywhere right now, so unfortunately. So there are different areas, but even with global warming, it's not just the cold, it's the freak weather that comes with it that can be damaging to these crops.
So for the grapevine, there's the acclimation process, getting ready for winter, but then there's also the risk of deacclimation too early and then having some frost early spring. Would it be applicable that you could potentially spray it on and keep it within the cold hardiness for a longer window?
Yeah, so it would be nice. Jim would be better positioned to answer that, but definitely his chemical does exactly that. So it protects, it seems to lengthen the dormancy on good years a couple of weeks. And was this the best we can do? I don't know. But it looks like the chemical is protective against against the buds developing, in February, because we have warm weather. You're wearing a T-shirt and all looks great. So it looks like it works for that. And we don't know all the details of it. Part of the work that I would like to do with Jim is to bring the system in grow chambers. So on that grant, if it crystallizes, we can refurbish some of the grow chamber here with temperature that go below zero, so freezing. And then we can do much more trial than outside. Outside, you're just at the mercy of the weather. I'm not saying it's not important, but I think we could crank up more data by playing with the temperature indoors. And then once we have a good idea, then we can see outdoors as well. But yeah, this chemical seems to be playing all along the dormancies and extend it at the far end.
Lily, you talked about doing the genetic kind of mapping, looking for the marker for cold hardiness, and then being able to use that potentially for breeding. Could that be used for other qualities that are desirable for grapevines? Because a lot of the issue with grapevines is that you can't have fruit development your first year, so you have to wait for the third leaf. So that extended period, if you could potentially take a young cutting, get the genetic information, and have an idea of what the grapes would look like down the line, you could accelerate that process.
Yeah, it's possible. What we're doing would not address that at all, but the technology we're using could be used. We would just need different cultivars, different controls, and so on, and really think how to tackle that problem. But yeah, at the end of the day, everything is governed, that's my point of view anyways, by gene regulation. And if not gene regulation, some epigenetic level, but it still translates to gene regulation. And it's the easy path to look at things today, and the costs are coming down. You see, I spent 3 years purifying a transcription factor. With a map, with transcriptome data, you would not do that anymore. And I think you can get some answers. The important thing is that if you want to use a map, really, you need to be able to cross. And then so you use something that behaves well, something that does not. You cross, you look at your F1, and then you start associating phenotype to genotype or to transcripts, and then try to walk your way through that.
Well, thank you so much, Charles. And here's a little thank you on behalf of COVI. And for those joining us online, the lecture series will wrap up in the coming weeks with a presentation from COVI professional affiliate Wendy McFadden-Smith. We're just working to reschedule that with her. It was postponed from last week, but we will keep you posted with that rescheduled date. So thank you for joining us.